Hi, my name is Ellen. I'm an undergraduate at the University of Kentucky, and today I'm going to be presenting about the topic of the relationship between the ventral nerve cord and specifically the A6 ganglion of the ventral nerve cord in crayfish to the hindgut of the crayfish, looking at neuromodulators using for stimulation. As you can see, there's a poster hanging behind me. I'm going to be using a kind of different format from the other videos that have been posted so far, but we're just going to see how it goes and see if the format works or if it doesn't, and then see how it goes from there. Now, beginning with why the crayfish was chosen as the subject of this study. The crayfish hindgut has been researched as a model for over a century, but it still offers potential for investigating the generation and regulation of peristaltic rhythms, including the mechanisms at the level of neural circuitry and ion channels. Like the vertebrate GI tract, the crayfish hindgut has intrinsic pacemaker activity. However, it's not composed of smooth muscles like the vertebrate GI tract, but instead striated muscles with gap junctions. The crustacean hindgut contracts spontaneously following denervation, but such contractions are typically weak and disorganized. Peristaltic movements require coordinated motor output from the central nervous system and seems to originate at the 6th abdominal ganglion. Now, previous studies have investigated the responsiveness of the crayfish hindgut to direct application of neuromodulators, and it's been demonstrated that application of serotonin and dopamine to the GI tract results in increased frequency and strength of contractions. Serotonin and dopamine are highly concentrated in the central nervous system of the crayfish and as well as the GI tract, and are directly responsible for peristalsis and muscle contraction. Dopamine is reported to modulate nearly every invertebrate physiological process studied thus far. Among the targets are peripheral organs, sensory organs, and the central nervous system. Our aim with this specific study was to investigate the influence of the central nervous system and ventral nerve cord on the innervation of the hindgut of the crayfish. We measured the force and frequency of the contractions after application of neuromodulators including dopamine, serotonin, and dopamine on the whole chain of the abdominal ganglion, and then specifically to the 6th abdominal ganglion. We then assessed the effects of the neuromodulators directly applied to the hindgut when isolated from the ventral nerve cord. The three main questions we wanted to address with this study are: 1. Are the neurons within the A6 ganglion susceptible to neuromodulation? 2. Is there descending control within the ventral nerve cord in addition to neurons within the A6 ganglion that can be modulated to alter the frequency of hindgut contractions? And 3. Upon driving the neurons to the hindgut, can the contractions of the muscle be entirely blocked by the presence of CO2 containing saline, thus blocking gap junctions by intercellular acidification? Now, taking a look at the method used, the experiment measures the influence of select neuromodulators on the activity of the crayfish hindgut when applied to either the A6 ganglion, the ventral nerve cord through ganglions A1 and A6, and then also directly to the hindgut. The neuromodulators used were including dopamine, serotonin, and dopamine. Each neuromodulator was administered at a 1 micromolar concentration in crayfish saline. This chart here breaks the method down into two different experiments. With experiment 1, we apply the neuromodulator first to specifically the A6 ganglion and nothing else, and we record it for 20 minutes, and then after that, we take the same neuromodulator and apply it to directly the hindgut and record again for 20 minutes. Now you can see that we did it for dopamine, serotonin, and dopamine. And each experiment is done for 5 times. And then we move on to experiment 2, where you apply the neuromodulator to the ventral nerve cord from ganglions A1 to A6, and then after 20 minutes apply this neuromodulator directly to the hindgut and again record for another 20 minutes. Broken down into steps, first we begin with the preparation, where we dissect the crayfish, exposing the GI tract and the A6 ganglion and the ventral nerve cord from A1 to A6. And in a side guard dish with crayfish saline, we secure the tail fins and the GI tract with pins using a side guard platform and Vaseline. We create a well to evaluate and isolate the A6 and the ventral nerve cord away from everything else. We then apply crayfish saline to isolated A6 ventral nerve cord. The second step is using a force transducer. We use a plus or minus 2 gram range sensitivity force transducer from AD Instruments. We attach the GI tract to the force transducer with both the GI tract and the A6 ventral nerve cord isolated in saline baths. We measure the force and frequency of contractions using this force transducer. Acquisition of 2 kilohertz and sensitivity of plus or minus 2 grams, collection for 20 minutes. Using a Kim wipe, we then remove the saline solution from the Vaseline well and add select neuromodulators to the Vaseline well and continue recording for 20 minutes. And then the final step is using a large pipette, we remove the saline solution from the dish and apply select neuromodulator to the GI tract and continue recording for another 20 minutes. Here are some photos of what I just discussed. This is the A6 ganglion that is directly connected to the hindgut. Here is a picture of a crayfish preparation that's been stained with methylene blue. This prep shows the GI tract and the A6 ganglion. Here's a larger picture of the whole prep, and as you can see, the telson is still there, and reaching straight up from that is the tan GI tract, and kind of off at an angle to the right is the ventral nerve cord stained with methylene blue. And here's a picture of the force transducer we use to record the frequency and force of the contractions of the GI tract when the prep is exposed to saline and neuromodulators. Here is an explanation of the calibration we use to calculate the difference from the force transducer. The first dip in the chart is after attaching a 2 gram weight to the force transducer, and the second one is a 1 gram weight. Originally, the force transducer is at 0 before the weights are assembled, and then the brash lines you see are a result of handling when the attaching and removing the weights. Here are some sample results of the data we gathered. While every time we did an experiment, we repeated it 5 times with each neuromodulator and applying it to the A6 and the hindgut, or the A1 through A6 ventral nerve cord and then the hindgut. But here we just have a couple of examples. In this case, this one is for optopamine applied to the ventral nerve cord and then applied to the hindgut. This is the display that we see on the computer that's connected to the force transducer. Here is the first 20-minute increment of time. This is like the control, where no neuromodulator, or no optopamine in this case, has been applied to the prep. Then this middle section here is after the optopamine has been applied to the ventral nerve cord. And then this last section is after the optopamine's been applied to directly to the GI tract. Now the results underneath it display the average number of contractions for that 20-minute time period. As you can see, the saline had the lowest average of the contractions for the 20 minutes, and when the dopamine was applied directly to the ventral nerve cord, it had about a little over 15, and then when it's applied directly to the GI, it's getting up closer to 20. Next to it, we also have another example. In this case, it's serotonin applied to the A6 ganglion, and again, the first time period is the control without any serotonin on the prep. The second time period is after it's been applied to just the A6 ganglion, and then the third time is when the serotonin's been applied directly to the hindgut. And then here again the graph shows the same sort of consistency, where saline has the lowest with about just over 10 contractions per the 20 minutes, and the A6 has around 12, and the GI has a little over 14 average. Then we also have another example. It's just an example with the dopamine with the A6 with the same sort of time frames, and then the same trend is shown also with a dopamine. Here we see a summary of our results shown in percent change. The red ones, the red dots on the graph, are the A6 when the optopamine in this case is applied directly to the A6, the percent increase in contractions. So this is actually a comparison between the number of contractions under just saline versus the number of contractions when optopamine is applied directly to the A6, shown as a percent change. The blue is also like this. It's a representation of when optopamine's been applied to the ventral nerve cord. So it's again that comparison, a percent change between just the saline and then going to optopamine on the ventral nerve cord from ganglions A1 to A6. Now, each dot is actually an individual shape, as you can see. So this is like a star, and that's an octagon. This is a cross. Each of these shapes represents a specific trial of 1 out of the 5. In this case, you can probably only see 4 dots, but this dot here is actually 2 separate trials that are so close that they kind of overlay on one another. The green is showing the percent change of when the optopamine was applied directly to the GI tract. And this is not a comparison of percent change between the saline solution and then application to just the GI. It's actually a percent change between either of these to that new step. So in this case, if you look, you can see this square, and it's a small percent change from just the saline solution to the optopamine on the A6. But then that same square moves up to almost a 50% increase in contractions after the optopamine's been applied directly to the GI. So if you remember, the actual method is application of the neuromodulator first to one of these and then to this. So that is why the visual representation, it goes from, it's like builds on one another from this step to this step, because this isn't going directly from saline to GI. It's going from saline to one of these and then just to this one. So with that in mind, even though the symbols seem to be relatively close, or like these down here seem to have a less percent increase in contractions than these symbols for the ventral nerve cord, that's not necessarily the case, because these are building on what is represented over here in these 2 rows. Here is another summary of the results, this time for exposure to serotonin. Again, the red is exposure to just the A6 ganglion, and the ventral nerve cord is blue, and the GI is green. And it's the same case where this one is comparison from saline to serotonin on the A6, and this one's saline to serotonin on the ventral nerve cord, but this one is the percent change from one of these to this one. So as you can see in this case, this large dot has again very little percent increase in contractions when going from saline to A6 exposure to serotonin, but its percent change increases a little bit in the next step still. If you want to see one with a lot of change, a percent increase of contractions, this dot on the ventral nerve cord has almost 100% increase in the number of contractions over the 20-minute period after the serotonin's been applied to the ventral nerve cord. And this dot correlates with this dot here, which again shows a very large increase in percent change of contractions, a little over 80% increase. So as you can see, for this specific trial, a large increase was seen both times under both conditions of adding serotonin to the prep. Here is the summary of results for exposure to dopamine, and again follows the same pattern of red A6 versus the saline, the blue ventral nerve cord versus the saline, and then the green GI against either the A6 or the ventral nerve cord, lining up the shapes to compare one another. In this case, there are two trials that the percent change in contractions was so large that the dots are actually not included on this first chart, and so there is another smaller chart to represent the outliers. And as you can see, the one blue dot showed a change in contractions of over 300%, and then one of the trials showed a change, I guess that same trial showed over a 450% increase of contractions after it was applied to the GI. So in conclusion, all three neuromodulators, serotonin, octopamine, and dopamine, showed an increase in the frequency of peristalsis and the force of contraction when it's applied to either the A6, the ventral nerve cord from A1 to A6, or the hindgut. With each neurotransmitter, there was a gradually increasing trend in both force and frequency, and direct application of these modulators to the hindgut has the most pronounced action, as evidenced by the bar graphs of the three representative samples. It should be noted here that the summary of results that I showed you discussed only the change in frequency of contractions that we saw after exposing either the A6, the ventral nerve cord, or the GI to the neuromodulators. But because we haven't finished compiling all the data for the change in the force that has been seen. But we can see that there is a change in force just by looking at these samples that we see from the computer. Because what we're counting with the frequency is just each individual peak that you see. Like in this view, it's just basically a line coming down. But the actual length of that line coming down is the measure of the force for that specific. So while we haven't gotten the exact number results for the change in force, we can see by looking at each of these and then the others that we haven't put up as examples that there is a change in force after the neuromodulators have been applied. Future interests we have following the same subject is the possibility of using a preparation where the hindgut is intact and examining the cerebral influence as well as the effect of neuromodulators on the central brain. Another consideration is varying the concentration of the neuromodulators applied, specifically to 300 nanomolar, for a comparison to the other studies that have been done already in the field. And Then examining the effects of receptor desensitization with high or long-term exposure to neuromodulators, examining serotonin and dopamine receptors subtypes on the hindgut within the ventral nerve cord and that impact motor neurons innervating the hindgut. Also, examine the neural input to circular contractions of the hindgut. These are the references of other studies that we looked at. And then we also should acknowledge the Science Outreach Program for undergraduate research apprentices at the University of Kentucky.